Now we are continuing with the same chapter, that is experimentation, and here we are discussing about two different chapter with different topics, that is what is mean by experimental requirements and how can we document an experiment. So let's move. Experimental requirements. We know that for doing an experiment or after an experiment is over, we get data. How these data are generally collected? Some data are generally collected by direct observation with a sense organs and by means of instruments. Okay, so they are generally classified into 5 different categories. For observations, we are generally depending microscopes as well as binoculars. That we all know that. Then if we want to measure the data, we are generally depending pipette, burette, hemocytometer. Yes, it is generally used for counting RBC cells in biological experiments. Okay. Then if you want to analyze any chemical as well as the physical parameters, we are generally depending colorimetry. You may be knowing that what is maybe colorimetry for qualitative analysis of a chemical compound or chemical thing. For example, chemical analysis of hemoglobin. Analysis of hemoglobin, we are depending this colorimetry. Spectrophotometry. Spectrophotometry, we are a lot of uses are there for spectrophotometry. By means of absorbance, we are able to determine the concentration of a solute from a solution. Then pH meter, we can we can identify the acidity as well as the alkalinity of a particular thing. So these are physical as well as the chemical parameters. So we can define this by means of this devices. Then processing. If we want to centrifuge a thing, and if we want to boil or stirring a solution, we are generally putting them in centrifuger or water bath. So for processing, we are generally depending centrifuge as well as water bath. But some modern biological experiments, we are mainly depends upon a highly advanced instruments. For example, PCR, polymerase chain reaction. We are generally doing this experiment in PCR machine. Then DNA sequencer. If we want to analyze the nucleotide basis of a DNA, we are generally depending DNA sequencer. Then if we want to do electrophoresis like agarose gel electrophoresis for DNA and RNA resolution, for DNA and RNA identification and also protein estimation, we are depending PAGE, that is polyacrylamide gel electrophoresis. Then UV transilluminators for visualizing these macromolecules. Okay, so for all these purposes, we are mainly depending upon the instruments. So these are instrumental requirements. So we can classify the whole experimental requirements into these 5 major categories, right? Then how can we document the experiment? A careful documentation of all the details of an experiment and reporting all these details are the integral part of the high quality research or high quality work. Okay, so for documentation of an experiment, we need to keep either a logbook or notebook. There's a difference between these two. What is maybe a logbook is that it is a private diary of all the daily activities of a concerned experiment. Okay, in the case of notebook, this is also a daily workbook, but it is mainly based upon the research project. Okay, it includes the research, experimental setup, data collection, explanation, procedures as well as result. Alteration in the notebook cannot be changed. Okay, so that's all about the documentation of experiment. In this manner, we are generally documenting all the details. Okay, then a scientific notebook serves as an certain purposes for future references. What are they? It generally, a scientific notebook generally covers the complete coverage of the whole work for a scientific paper. You may be knowing that in journals you can access the different kinds of scientific papers. So this scientific paper is actually a complete coverage which is taken from a scientific book. Notebook, either a notebook or a logbook, and we can make suggestion for repetition of that experiment. If a work seems to be invalid, we can make a suggestion for repeating that experiment once again. And sometimes most of the time it generally provides valid evidences. And this scientific notebook generally forms a solid base for the evaluation of work. Okay, so that's all about the documentation of the experiment. I hope you understood. If you have any doubt, please do in the comment section. Thank you.